we might get started. So welcome everybody to our webinar on TB genomics. I'd like to start by acknowledging the traditional owners of the lands from which we're all joining today. I'm here in Meumann, Brisbane, on the unceded lands of the Turrbal people, and I'd like to pay my respects to elders past and present, and welcome any Aboriginal and Torres Strait Islander people in the audience joining us today. So this seminar has been organised by ACTnet, the Australian Clinical Tuberculosis Network. If you're interested in receiving our newsletters and checking out our podcasts, you can head to the website, which is actnet.org.au. So my co-chair for today is Professor Justin Denholm, who's the Medical Director of the Victorian TB Program. He's also an infectious diseases physician and ethicist, a staff specialist at the Royal Melbourne Hospital, and a clinician researcher at the Doherty Institute. I'm Ella Meumann. I'm an ID physician, recently finished my PhD on using genomics to understand the epidemiology of several pathogens in the top end, including Mycobacterium tuberculosis. And I'm currently doing further training in clinical microbiology at Sullivan Nicolaides in Brisbane. We've had a slight change to the program today. So Dr. Narelle Sherry is unfortunately unable to join us. However, we're very lucky to have Dr. Kristy Horan stepping in at fairly short notice, and I'm sure she'll do a really great presentation. First, so questions. I think you should be able to put them in the chat, and we've had a few minor technical issues so far today, but hoping that Justin and I will be able to keep an eye on those, and that there'll be a few minutes at the end of each speaker's presentation to ask a question or two. Our first speaker today is Dr. Chris Coulter, who's a microbiologist and the chair of the TB Expert Advisory Group for Tuberculosis in Queensland, and the director of the Queensland Mycobacterium Reference Laboratory. And he's going to be giving us an introduction to TB genomics, and I'm hoping he'll be able to share his screen and get started.
Yep. Thank you, Ella, and I'll just fiddle around here to share the screen. Just need to put it into a different format. You probably can't. Is it on the wrong format?
It's not currently in presenter view.
Is it now?
No.
That's a shame. How about now?
Yeah, perfect.
Okay. All right. Without further ado, we'll get going. So, thank you to ACTnet for asking me to speak and to introduce and set the scene regarding this symposium on TB genomics. So, thank you for the kind introduction, and for my sins, I'm also an ID physician, and that side of things, I work at the Metro South Clinical TB Service, seeing a few patients. So what do we want from the laboratory as clinicians? Well, when we receive a sample, clinicians want to know, is there mycobacteria in the sample? Particularly, is there TB present? And if it is TB, which we initially identified at complex level, what species in the complex? Then we want to know, is this strain of TB susceptible or resistant to our key first-line drugs? If it's resistant, what about the second lines? But from a public health perspective, and also from a infection controls perspective, we're also wanting to know, is this likely to have been transmitted from another person? Is that transmission likely to be recent? Can we identify clusters and outbreaks? Many people may not realize that laboratory cross-contamination can account up to 3% of TB strains in a laboratory. So it's very important for patient safety to identify that. We want to know if reinfection is due, well, if infection is due to relapse or reinfection, and lastly, has treatment been successful? So I won't go through all the processes, starting from sputum receipt and processing, rapid molecular detection, culture, DST, and genotyping. But for a drug susceptible strain, this might be as short as 21 days, which is a lot longer than it would be, say, for Staph aureus. And for a drug resistant TB, it could be over 3 months, up to 100 days. So our existing methods can lack sensitivity, specificity, for typing, discriminatory power. They can be slow, costly, or all of the above. So just a quick, very brief, I should have said very brief history of sequencing. So Watson and Crick and their other Nobel Prize recipient, Maurice Wilkins from New Zealand, didn't invent or discover DNA, but they did, particularly Watson and Crick, deduce the three-dimensional double helix structure of DNA. And they couldn't have done it without Rosalind Franklin, who's the other photo, who took the crystallography photos of which their deductions were based. But unfortunately, she's not often remembered and was excluded, or not, she was not awarded the Nobel Prize for her work. So this is a nice little diagram from a recent review on the history of sequencing. And there's just a couple of things to point out. First of all is that this work here, which was the sequencing of a phage X174, was performed by Fred Sanger and colleagues, and it was the birth of chain of termination DNA sequencing, what we commonly call Sanger sequencing. And that set the scene for the next few decades, particularly involving cloning with Sanger-based shotgun sequencing. And then the next key point we get to as TB doctors and scientists is 1998, when Cole and colleagues published the complete genome sequencing of M. tuberculosis. The other thing to draw from this diagram is as a scientific discipline, progress from early sequencing methods to these Sanger-based shotgun sequencing, then we get into the next generation sequencing. And then when it comes to single molecule sequencing, such as we see with Oxford Nanopore or PacBio, some people refer to the term third generation sequencing. So this would have been a good paper to get on because it's had a lot of citations, and it told us many key things about the M. tuberculosis genome. It used the H37RV strain and deduced it was 4.4 million base pairs long. In 2013, a resequencing investigation added only 4 base pairs to the original sequence. So it codes for about 4,000 genes, and a little bit different to a lot of other bacteria. Its GC content's quite high, around about 65%. And up to 10% of the genome is characterized by repetitive proline glycine-rich sequences, which from the sequencing point of view can be difficult to map. So what actually is next generation sequencing? So in contrast to all that went before it, it's faster with a higher throughput. And the key phrase here is massive parallel sequence reads, whether it be DNA or RNA. And compared to, say, the billions of dollars spent on the human genome through shotgun Sanger sequencing, we're now talking relatively low cost. So what can next-gen sequencing do for TB medicine? Well, we can provide rapid confirmation of that a Mycobacteria is TB and what member of the TB complex it is. Rapid detection of antimicrobial resistance when we consider those long lag periods for phenotypic DST, and potentially a broader menu of AMR detection, not necessarily limited by what we can do with a test tube. We can tell relapse from reinfection and, as I already intimated, lab cross-contamination. It allows precision genomic comparisons for public health and infection control action, and overall shortens time for complete results. including typing. And if we use next generation sequencing on a specimen rather than culture, rather than 2 to 4 weeks, go to woe, we're looking at maybe 1 to 2 days. And from the developmental point of view, it provides key information for R&D of new drugs and new vaccines. So let's talk about the process of whole genome sequencing in a little bit more detail. We conveniently divide it into the wet lab processes and the dry lab processes. And the dry lab is really where all the bioinformatics occurs, and the wet lab is preparing the DNA and sequencing the DNA. So let's talk about wet lab first. This was Stradbroke Island on the weekend. So first step is to DNA extract and purify, and there's a number of methods of doing that, as probably many of you know, starting with cell lysis and purification, and that can be automated for higher throughput activity. Now, a number of stages of the WGS process, it's really important to make sure that there is quality DNA, and that starts off with what goes into the library preparation. And I won't go through the whole slide, but to particularly highlight DNA quality, which can be assessed spectrophotometry wise, and DNA quantity. And there's a number of ways of doing that, for example, the qubit system. So there's a number of different platforms. Longer read sequencing is the properties of Oxford Nanopore and PacBio, and shorter read sequencing, particularly with Illumina and Thermo Fisher. And in general, the advantage of shorter reads is less errors, although PacBio has a low error rate as well. So we talk about Illumina sequencing, but in fact, we talk about Holden cars, there's quite a lot of them. And there's quite a number of options in the Illumina suite of instruments. The most recent one is the NovaSeq X, announced at the end of last year, which has new chemistry. And the other thing that's caused a little bit of a stir on the Twitterverse recently is that Illumina have announced their chemistry for complete long read sequencing, and this uses their instruments such as the NovaSeq X to produce long reads. I don't know the full impact of that, but I gather people are excited. This is a schematic of the Illumina sequencing by synthesis strategy, which uses bridge amplification PCR to form clusters, and then these are sequenced. And to give a full overview of that would take time, but I advise you to go to the Illumina website and look at the video cartoons, which really do a much better job than me in explaining the actual sequencing process. But the end result is, depending on the Illumina instrument you use, paired 150 or sometimes 300 base pair long reads. And what's the output of all this? Well, the output is a FASTQ file. And this is just a little example of a snippet, a mere fraction of a FASTQ file. They're very big files. And once we've got that, we move into the bioinformatics, the area of the dry lab. So first thing, again, quality checks. And there's a program commonly used called FastQC, and it looks at per base sequence quality. It looks at the overall GC content. For instance, with TB, if you found you had a GC content of 50%, well, you've missed something because it should be around 65. And it looks for duplications and overrepresentations of reads. One of the things you might have heard people talk about but may not fully understand is the Phred quality score. And this is an exponential scale of probability of an incorrect base call, and the inverse being the probability of a base call being accurate. And generally, we like a Phred score of being on 30 or above, where we've got 99.9% base call accuracy. And FastQC can graphically demonstrate that by plotting Phred score of reads against the length or position in the fragment's been sequenced. So on the left is some good quality data. Green's good, orange is maybe, red's bad. and you can see that in this trace here that we have everything essentially in the green good zone and high in the green good zone, and the confidence intervals are also remain in the good zone. And it's really only when the sequence gets at the extremes of its length that we start seeing some increase in base error. Contrasted to what's on the right, where this is a bad sequence and should be repeated. Then things need to be trimmed and tidied and filtered, and two common programs to do that are Trim-O-Matic and Kraken. And the idea is to trim out low quality regions, the beginnings and ends of reads and the Illumina adapters, filter out contaminants, so other organisms or DNA of other organisms, reads that are too short or indeterminate. And it's commonly, particularly with a short read strategy, it's common to exclude repetitive elements. That should be PE, not PPE. So proline glycine, proline glycine sequences and insertion sequences such as the IS6110, which are otherwise quite useful for diagnosis, and the now obsolete RFLP. While one can do de novo assembly for the practical purposes of TB diagnostic labs, it's assembly by mapping. So this is mapping to a reference genome. This can also sometimes be referred to as resequencing or comparative assembly. The reference genome is commonly the H37RV, but sometimes it might be useful to use an inferred ancestral TB genome. And some of those questions that I asked before can then be answered from the successful mapping strategy, such as what species, what's the lineage, and what are the SNPs or single nucleotide polymorphisms. So just a reminder that it's not just one lineage of TB, there are many plus animal strains as well. So you note that M. bovis is not on this. It's now, I think, up to 9 lineages with lineage 5, 6, and 9 from Africa. But the 4 common ones we see are East Asia, which is lineage 2, includes Beijing, and then depending on where you are, varying proportions of the other strains. But back here is where we would be thinking about an inferred ancestral strain. So the comparative method of assembly, we have whole millions of with Illumina short fragments, and we're going to map them computationally to a reference genome. There will be some areas perhaps where there is no mapping, so that's a lack of coverage in those sections, and there may be reads left over that are not found in the reference genome. I'll use the term coverage. So coverage breadth is the fraction of the genome sequenced by at least one read. Then coverage depth is looking at individual base points and working out how many times that base has been sequenced. And in the diagram on the left there, you can see that there's 2 fragments that have that base in question, whereas we get to move to our right, we can see up to 9 fragments with that base in the sequence. The other term which is worth knowing about is uniformity, which measures the evenness of the depth coverage across the genome. So we want to look for variants or SNPs. And so just in a simple fashion, we here down the bottom in orange, we have the reference genome, and we use software and visualization tools to look for exceptions to that. So this test strain in every sequence read there is the same, except at one point, at this point here where a C has become a T, and that is a SNP. So just to recap, we have our sequence reads, which are FASTQ files. They're mapped to a reference, and then we're now talking about BAM files, and then we're talking about variant calling, and we can then produce another type of file called VCF or variant calling files, and that's a little snippet of one down in the right-hand corner. But we need to, the variant files and BAM files can be viewed using viewing software, and this is Artemis, which is a Sanger-based open access software that allows viewing and SNP annotation. So SNP annotation takes us from saying we've got a SNP to where is the SNP, what amino acid code, does it change or not change if it's a synonymous mutation? And whereabouts in the gene is it? And then we can make inferences about whether that's likely to be significant or not. Some common mutations in, say, the RPOB gene, we know for sure that's going to confirm resistance, but there's lots of mutations that might be discovered in a whole genome. How do we know they're significant? Well, one useful document produced in June 21, or published in June 21, is the WHO catalogue of mutations. And this was derived from the Cryptic Consortium, which is largely European and North American based, but I think Vitali's laboratory also contributed to the Cryptic Consortium, and there are other bodies involved that basically provided paired whole genome sequencing data and phenotypic drug susceptibility data. Only Illumina data was considered, but there was 38,000 genomes to be able to provide information for the catalogue. And if you go to that catalogue, you'll be able to look up the drug, the mutation, sensitivity and specificity, grading, and after some sort of expert contribution, a final confidence grading, whether, so something like serine to leucine at position 450 in the RPOB gene, that's your most common rifampicin resistance conferring mutation, clearly associated with resistance. And if you take the time and look at the numbers, you'll see why. Whereas this one down the bottom at position 695 seems to have no bearing on resistance. Despite those 38,000 strains, when the quality rules were applied to mutations that putatively could be associated with bedaquiline or clofazimine, we actually came up with no mutations that we could clearly grade as being associated with resistance. Now, the group that oversees this is meeting at the end of this month, and the data will be presented to the review group, and hopefully will be informed by a lot more information since the use of bedaquiline and clofazimine, which are now key drugs for treatment of drug-resistant TB. We're not reporting in our lab for diagnostic purposes whole genome sequencing, but this is an example of a research-only one. And you can see that there's some parameters at the top of the report about the specimen, about the sequencing, some conclusions about whether there's drug resistance, stating what the lineage is, and then whether there's likely to be drug resistance. And this example given, this is an XDR strain with all four first-line drugs are resistant: moxifloxacin, ethionamide, bedaquiline, clofazimine, all with genotypic resistance. And lastly, we're interested in demonstrating relatedness, and that there's a number of different ways. It might be a pairwise SNP matrix, where you're looking for the number of SNP differences between two strains. It might be a phylogenetic tree, of which there seems to be, I was going to say infinite, but that's not true, but there are many ways of drawing a phylogenetic tree. But does that tell us about transmission? Transmission is more complicated than just saying two strains are the same. So some of the things we consider are the timing of the sample collection, symptom duration, and period of infectivity, and the two are not necessarily the same. Of course, genetic relatedness, but we might have to account for missing cases if we're trying to analyze a putative outbreak or cluster. We might have to account for within-host variability of genetic relatedness, and putting all those factors together, come up with a model that suggests the most plausible directions of transmission. And one that we've been using in our lab, and when I say we, I mean Arnold, is TransPhylo. And if I've got a couple of minutes, I just want to mention targeted next generation sequencing, mainly because it's enticing, because it can use a clinical specimen rather than a culture, and because there's a commercial method recently released called DeepPlex. And this targets, it's basically the same process as whole genome sequencing, except it has a multiplex PCR step specifically aimed to amplify the gene targets in question. So the Deeplex looks at 19 genes, 13 of them are covered drug resistance. The other one looks at things like mycobacterial identification, so it can be used for NTM ID as well. It's not as discriminatory as whole genome sequencing for looking at relatedness, but you can go a reasonable way and certainly look at excluding relatedness with oligotyping, and it also tells the lineage. One of the things to be aware of about anything that works off clinical specimens is that you get a lot more human DNA and a lot more contaminant DNA to contend with. And how this would work is it could be in the field setting that you do a multiplex PCR, and then the products of that PCR go into a standard Illumina benchtop or perhaps more investigationally Oxford Nanopore. And all the bioinformatics really takes place not in your lab, but you upload the sequence to the Deeplex web app, and then it comes back with the answers to the questions that were indicated on the previous slide. So in summary, whole genome sequencing offers substantial advantages to our current methods. Targeted next generation sequencing is attractive, particularly because of its ability to use specimens. Not that people are not trying to do a whole genome sequencing on specimens, and there's been varying degrees of success, but it's not common practice. And lastly, I think people would be aware that the computational data requirements are enormous, and how best to store, retrieve, and use the data is still being refined. And just to acknowledge that some of the images I've reproduced from this free and online course, nicely entitled Mycobacterium and Tuberculosis NGS Made Easy. Tim Walker and the SRL network for some of the images on the mutational catalogue, and some material from the Sanger Institute website. So thank you.
Great, thanks very much, Chris. Now, I'll take the opportunity for us to just mention that we'll have a brief moment for questions after each of the three presentations. We may have some time at the end for some general chats, but also this is an invitation just to say that as everyone is speaking, please feel free to put questions into the chat, and we'll do our best to feed them through. Now, I see a note in my panelists chat that there's a hand raised from one of the attendees. Tanya or others, just so you know, we can't see the attendees. So when we're in host and panelist mode, those will just need to be fed through to us. But putting them through the chat function will be the best way for us to be able to read them. So at the moment, Chris, I don't have any questions, possibly because I can't read them. But what I might suggest is that we're at 1:30, and so why don't we move on to the next presentation? In the meantime, if you do have questions that you'd like to feed through to Chris as well, we'll take questions for both Chris and for our next speaker, and then perhaps have an opportunity to come back and talk about those afterwards. So Chris, just thanks again for a fantastic presentation and a great start to this really interesting seminar.
Thank you very much.
Now, I've got the pleasure of introducing the next speaker, Kristy Horan. So Dr. Kristy Horan, very pleased to have you with us today, Kristy. Kristy's bioinformatics section leader at the Microbiological Diagnostics Unit, Public Health Laboratory at the Doherty Institute, which is also where I'm based. So I also have the pleasure of having worked with Kristy around some of the really fantastic work that she does in TB whole genome sequencing, and particularly around the public health implications in Victoria. So Kristy, I will hand over to you. Really looking forward to your talk, and then pop back on with any questions we might have at the end.
All right, thanks heaps, Justin. Let me just get myself organised. Make sure we've got all the things going on. Okay. Can everybody see a presentation? I really hope you can.
You can. I've got you in the other mode. So if you can swap modes, I've got presenter mode up on my screen.
Oh. I don't know what that means.
Oh, I can see your slides, but I can also see your speaking notes in that format. So you're sharing your screen, but if you just hit presenter mode, then it should switch them between the two, I think.
Okay, sorry.
No, that's all right. Do you want to pop
Everybody.
it back up? And while Kristy's getting that settled, I'll just also say that I've got a few text messages from people offline to say that the chat function is doesn't seem to be working for at least some of our participants. We'll see if there's something we can do about reactivating that in the background, but my apologies. If not, it may be that sending, if chat is completely off, then there's nothing that you can do about that. But if, yes, we think it should be working now and turned on. And so if you can message the hosts and panelists, or in fact, if you can just message everyone with your questions, that'll be helpful, because then people can see what you're asking as well. And I'll keep an eye on that in the background. Okay, so back to you, Kristy.
Okay, well, I'm just going to have to do this the old-fashioned way and wing it, because I'm not smart enough to figure out presenter mode.
You've done exactly the right thing, and it's in presenter mode now, and we can see the presentation. So well done.
Okay, very good. All right, so thank you very much for the introduction and for letting me come along today and talk to you about the work that our team's been doing in the implementation of TB genomics for AMR. So this is very much a team sport. So I do acknowledge that the work that I'm presenting today is actually not just mine. We've got a whole raft of people that are involved. So yes, if I forget to mention you by name, I apologize in advance. So implementation of whole genome sequencing to inform or infer AMR is actually kind of challenging, which, I mean, who would have thought, right? And it's difficult for many reasons, but I'll sort of step through some of the major challenges that we have faced in implementing this technology for routine reporting of AMR inference from genomic data. So pretty much everybody on this call will likely be familiar with the concept of you have a phenotype, which helps you to infer a clinical outcome. Everybody's happy with that relationship. That's very well established. But what happens when we bring the genotype into the question? And so it is very difficult to relate a genotype to a clinical presentation or a clinical response. And unfortunately, it's probably unethical to test some of those genotypes in real life human beings. So what we do instead is we look at how does the genotype relate to the phenotype that we see in the lab. And this relationship, everybody's a lot more comfortable with. There's no ethical problems here. So we try to use the condition that we're comfortable with, which is the phenotype, that can then go on to infer that clinical response or that clinical relationship from the genotype. So we sort of use this as a intermediary or a proxy to make that link between genotype and some sort of clinical outcome. So how do we infer an AMR profile? It's kind of challenging. There's a few different ingredients, and I've got two of the, probably in my mind, the most important ingredients to infer an AMR profile. And the first one is the sequence. So here we need to have a good quality DNA sequence. We need to be relatively confident or highly confident in the accuracy of our sequence recovery. And our sequence recovery can be informed by both the quality of the DNA, but also genome biology. So some species is easier to do than others, and also the sequencing quality, as well as the species. So for TB, it's important to sort of realize that when we sequence a TB sample, we're actually sequencing a representation of what's present in the patient at the time that the sample was taken. So not like something like Salmonella, where we pick a single colony off a plate. With TB, we got a midget sample that goes onto a plate. And so we're sort of gaining that whole population, which is a challenge to interpret from a bioinformatics perspective, because we don't just have one population of data. So that's a consideration that we have to take into account. But the other consideration that we also have to take into account is our knowledge base. So do we understand the AMR mechanisms? And this is, Chris helpfully introduced everybody to the WHO catalogue, but we need to assess what's the suitability of that database as well. So some AMR or antimicrobial drugs are much better understood than others, even ones that have been in use for a long time. So the difference between rifampicin and pyrazinamide is striking. There's a few mutations that are, solid rifampicin inducing resistance. Pyrazinamide is a little bit trickier, a bit because the mechanisms that underlie pyrazinamide resistance are perhaps not as well characterized. So our knowledge of those genomic determinants we should be looking for is challenging. So there's both of these things, and we sort of have to assess all of this when we're looking to implement genomics and whole genome sequencing in a public health lab. So I've just spent the last few minutes telling you, that this is hard, and it is. It's not straightforward. But we kind of persist with this because whole genome sequencing does have a lot of benefits. So we have multiple uses. Again, Chris mentioned things like outbreak investigations, transmission networks, identification of sample mix-ups or lab contamination. For AMR inference, it's really powerful. We can identify all the known mutations, not just what is, so we can identify what is already known, but we can also have that data persist forever. It's like the ultimate renewable resource. We can go back and reinterrogate it for new information to help inform perhaps some of those trickier cases that come across our desk. And cost-wise, it's relatively low if you're doing it in a high throughput setting. So if you've got lots of samples, per sample, the cost is cheap, is relatively cheap. In addition to that, as I've put here in the figure, it also has the potential to reduce some of that time to reporting of an AMR profile in TB. So, as everybody here knows, culture from a midget can take, a week to day or days to weeks, onto a plate, weeks to months, depending on what you're looking at. Developing that AMR profile can be challenging and time consuming, particularly if you have a resistant sample. So, if we can take the samples directly from the midget or from the slope, we can alleviate some of that time difference. So I put this up here as a description of how we go about addressing some of the challenges that we face when implementing whole genome sequencing in a public health setting. So, some of, as I said earlier, some of those mechanisms that we don't know about, for pyrazinamide and some of those novel or newer drugs, bedaquiline, delaminate, etc., we don't really know what they do. And that's a really interesting research question, but I'm no longer in the business of interesting research questions. I actually need to be able to take what's known and report that in a way that is robust, transparent, and relatively easy to understand. So from the bioinformatics perspective, we need to understand what the biological question is. Now, that can be difficult for a bioinformatician because most of us aren't microbiologists, we're not clinicians. I never want to go back into the lab, never want to don a lab coat again. So we have to understand the biological question. We also have to have a good overview of what tools and databases and approaches are currently being used and how they may inform our developmental choices. We also have to validate it. So we have to be able to understand, with the process that we've developed, what can we do and what can't we do, more importantly. So we need to have a really good grasp on where our limitations are. So the biological question in this case is, can we identify genomic determinants? And in TB, unlike many other pathogens, the genomic determinants are purely SNP or small deletion based. So they're acquired changes, single changes to the genome, rather than acquisition of whole genes, which you see in a lot of the enteric pathogens. So we need to be able to identify single SNPs and deletions, which is hard. We also need to be able to interpret those, because once we've figured out how to identify them, we can identify all of them, but the question then becomes which ones are relevant. So we need to be able to interpret those relationships or those outcomes. Tools and databases. So there are many amazing TB tools out there already, but many of them don't actually address the public health and clinical reporting aspects. They are very much focused on that interesting research questions. So they're not suitable out of the box, as in, you download it, you install it, you run it. It doesn't really work for reporting for clinical health. So, we have had to develop additions, so wrappers, to sort of address some of those issues. And then the database. So many of the tools that are available, such as TB Profiler, the microbe tools, as well as Ariba and some other tools as well, they all have their own databases, which largely, I'm not going to lie, are pretty much the same. There's very little difference. And then we have the WHO catalog as well. So, you also have to assess those aspects. So what database is actually good in your setting? Then when it comes to validation, the tricky part here is you need to validate it against your gold standard. Now, when we're talking about can we recover genomic determinants, the problem there is what's the truth? So there's very few full genomes from Sanger sequencing PCR or another traditional technology with which to compare how our bioinformatics pipelines too. So we fall back then on in silico generated data. So we simulate mycobacteria data, and I'll go into that in a little bit more detail in a moment. We also do validate against PCR as well. But again, the availability of that type of data, which has both the PCR and the whole genome sequencing, is scarce. So it's not terribly useful. And then we have the inference of the drug resistance. So again, this is also limited by what is available. So what drugs are routinely tested for, that also have whole genome sequencing data. So pulling together a data set, that addresses that was a need that we had too. And finally, this is just on the right. I'm not going to go into detail here, but from a public health perspective, we also implement and then continue to verify. This is not a static process that's set and forget. This is a dynamic process. Tools are always being updated. Databases are always being updated. We need to make sure that we're providing the best possible analysis based on the available tools and the databases that we have. So we have to have document control as well as version control for all of our tools, as well as a verification strategy. So if you have any questions about that or you wanted to chat about it offline, I'm happy to talk about that all day. The other important factor here is discrepancy resolution. So no approach is going to be perfectly matched to your gold standard ever. Doesn't happen. But the important thing there is understanding why. So is it simply a data entry issue? Your gold standard was entered into a database incorrectly. Happens. Is there simply a bug in your code that you might fix up and then everything is fine? But sometimes there are issues that can't be resolved in that fashion, and they're fundamental biological issues or knowledge-based issues. So in those respects, we need to know what they are. So we can either decide to not report them, or we can decide to report them with a comment or a caveat to allow the end user to still consume useful data, but with an understanding of what the limitations of that data might be. So we really do need to understand where is the line and how many toes can we put across it. So we developed a tool that's really just a wrapper for those of you that are familiar with these sorts of things, that implements TB Profiler with a custom database that we've curated. We've called it TB Tamer. It's part of what I call the TAMER family of tools. We also have an ABRA-TAMER, but I won't talk about that one today. So yeah, it implements TB Profiler to detect variants, so both single nucleotide polymorphisms as well as deletions, and then filter based on resistance conferring mutations. And those resistance conferring mutations come from our custom database. We then apply some reporting logic to generate some tables with an inferred phenotype, so what we think the likely phenotype would be, and what that drug is, what the mutation is for that particular drug. These are then piped into our laboratory management system, which generates a per patient report that is fed back to the submitting laboratory. So in most cases, that is our Mycobacterium reference laboratory up at Vigil at the Doherty Institute. But we do also do this, I think, for other jurisdictions or other labs as well. So we generate these per patient reports. Now, the other thing that I'm not going to talk about today, but is also required, is this interpretation of genomic relationships. So at MDU, we have a NATA accredited SNP detection and relationship pipeline. And anything that we generate here or any sequences that we make also need to be suitable for input into that pipeline for us to maintain our NATA accreditation. So I've just put the GitHub down here. So if anybody's interested, just let me know later. That is not in the right order. Apologies. That's the right order. So sorry, I did these this morning. So when we're talking about inference of AMR, that is, in addition to our database, is heavily dependent upon our ability to accurately recover the sequence of the input genomes. So what we did here was we generated a data set of a simulated in silico data set of 200 genomes based on the H37RB reference with SNPs or variants introduced at specific sites so that we know where we've mutated. And then we went and we ran our pipeline and then checked how many of those introduced SNPs and variants could we recover, and did we have any that we got wrong. So overall, our pipeline had a pretty high performance. If we had a genome coverage, average genome coverage of greater than 20, so up to 40 times, we could recover almost 100%. Oh, so 99% at 40 times and 100% above 40 times of our introduced variants. And we had a very low, a very high PPV. So overall, our recovery of our genomes, I was after this pretty confident that we could accurately recover the input genome sequences. So the other thing I just wanted to mention was this idea of midgets. So in our setting, we have our Mycobacterium reference laboratory that do the culturing and the DNA extraction and all of the really hard work for TB. And then they send the DNA extractions down to the sequencing laboratory, and then that comes through to the bioinformatics section. So we would like to be able to do this process from midgets as well as from slopes, but our NATA accredited pipeline for genomic relationships was developed for information from slopes. So we have had to go back and re-verify that the information that we get from midgets is also adequate for both processes. So we had 66 matched groups of sequences from slopes and midgets that passed our sequence quality process. And of those, we had 62 where we identified exactly the same AMR resistance mutations. Now, I will note here that the proportion of reads for each of those mutations was different. So in the midgets versus the slopes, the allelic frequency was not identical, but we were still able to recover the same mutations in both. We had 4 groups or 4 pairs that were discrepant. 2 of those, we had multiple lineages detected, which meant in the midget, which meant that what we detected in the midget was potentially a mixture of 2 samples. And once they were plated out onto a slope and cultured, we could then only identify 1 lineage from that sample. So that is not necessarily a reason to completely discard those results, because the sequence we got from the midget was what it was, but maybe grounds for perhaps going back and reassessing those steps in the laboratory, just to make sure that nothing was mixed up, nothing was added that wasn't supposed to be added, and then down into the sequencing laboratory as well. So it would just be a red flag to say, hang on a sec, let's go back and have a look at this one. But note that these ones with multiple lineages would not be input into our phylogenetic pipeline. We also had 2 samples where we had mutations in the 23S gene in the midget that were absent in the slope sample. Now, mutations here are implicated in linezolid resistance. The 23S gene is notoriously challenging because it is very, very similar amongst many types of bacteria. So if there is some low-level contamination in the midget tube, we may sequence 23S that is not from a Mycobacteria tuberculosis sample, and subsequently it's close enough to align to our reference and we pick up the mutations. But then once you culture it out, those contaminants are sort of lost. So it's a challenge. We don't yet report linezolid resistance routinely, but this would be, again, a red flag for this is something that we would need to look at. We may not report these after further investigation. So our validation of our phenotype, our data set. Now, I'm not going to bang on about this too much because this has been done by others in some fantastic studies over the last few years, showing the relationships between mutations and phenotype. I just wanted to sort of demonstrate that the data set that we have developed here is primarily cultured at MRL and sequenced here in our molecular lab. And we did it that way because we wanted to be able to report things that were consistent with what had already been reported. So treating our MRL and our current sequencing process as our gold standard, can we meet that using this genomic inference? So our data set had about 3,500 samples in it. 2,500 of those were from Victoria. The remainders were pulled in from various public data sets. We had a good representation of the 4 major global lineages. We also had a few non-tuberculosis sensu stricto species included, just for specificity. We had a good representation of the first-line drugs, which is great. And then we had a sort of a bit rubbish representation of some of the second-line drugs as well. But this is to be expected because there's not that much data out there that has both the phenotype and the sequence for those drugs. So as things progress, these data sets will increase and we may need to go back and re-verify or revalidate. But at the time that we developed this, there wasn't that much out there. So first-line drugs, again, very consistent, equaling, if not exceeding, many of the published studies. Our first-line drugs, we were good. Our second-line drugs, moxifloxacin is the only one I've listed here in this table for the sake of time. But again, it had quite good performance metrics, again, equivalent to what we see in the literature. To our second-line drugs, as I said, moxifloxacin was quite good. Many of the others were also performed quite well. However, cycloserine and para-aminosalicylic acid did not perform well. So these are to be reported with a unvalidated comment to say that we can detect these mutations, but we have not been able to validate that they have any relationship to phenotypic resistance. So this is due to both the low numbers, so there's not a lot of resistant samples available, but also to, I think the databases are sparse in the mechanisms that determine these drugs. So using our validation data and our reporting requirements, we were able to develop a reporting logic that allows us to report drug-resistant profiles that are consistent with the WHO guidelines. So we report a susceptible if we see no known mechanism. We report monoresistant if we see a single mutation or mutations to a single drug. We have polyresistant. So this is where we have a mutation that is in more than one, but not rifampicin. And then we have monoresistance to rifampicin. So we only have rifampicin resistance. Polyresistant with rifampicin, multi-drug resistant, where we see rifampicin and isoniazid with another first-line drug, or rifampicin with another first-line drug. Interestingly, because of the lack of available information for bedaquiline, linezolid, we can't distinguish between pre-XDR and XDR because the definition is pre-XDR has fluoroquinolone and it is an MDR, and extensive drug resistant is that plus. And because we can't see the plus, we don't know if it is an XDR. So we sort of report those as a group and obviously recommend phenotypic testing correlation as well to sort of distinguish between the two. So how do we report these? So when we were deciding how to report, and this is just in general, but I'm applying this specifically to TB here, is who's going to read these reports? And in our case, there'll be medical microbiologists reading them, clinicians reading them, epidemiologists reading them. So we need to sort of figure out what is useful to those stakeholders. What is it that they are going to want to see? So we report the presence of mutations or no mutation detected. We also report the inferred antibiogram for TB, as well as the drug-resistant profile. There was a lot of conversations and discussions around how much detail to involve, how much detail to provide. These reports need to be obviously informative and transparent and provide sufficient information. But if I gave you a table of all the mutations we found, I mean, that's not terribly meaningful. So what we elected to do was provide information about the level of resistance. So whether it's low level resistance predicted, resistant predicted, or whether it's susceptible, and what confidence we have that the mutation identified infers the level of resistance reported. So that was the level of detail that we send through. So this is my last slide, I think. And this is just to reiterate that this is very much a team sport. The system that we work with here in Victoria, which I think is similar to many of the other jurisdictions that have the capacity to have developed something like this as well. You know, we want to be able to capture and communicate additional mutations of interest. So the pipeline that we have actually captures much more than it reports. So it captures many of these unvalidated genomic variants for prospective monitoring of some of these unresolved phenotypic observations. We're also, personally, I am also particularly interested in some of the PPE genes, where there are some studies that demonstrate that they are linked to pyrazinamide resistance, but there's really insufficient data to say that that's actually the case in reality. So, you know, monitoring these things prospectively. So we're sort of setting up a system to allow us to do that. And we also engage in regular stakeholder meetings. So we have regular meetings with the medical microbiologists, bioinformaticians, as well as the microbiology lab experts to discuss and resolve any discrepancies that we see, any challenging cases that we have, sort of take them on a case by case basis. And we also engage fortnightly with the TB program and the Victorian Department of Health to discuss the genomics. So this is both transmission networks, as well as occasionally some of the more interesting AMR cases as well. So with that, I would just like to acknowledge the team. It is a big team. So in particular, Narelle, who is a massive driving force behind this particular project and the development of the genomics program for TB. The rest of the bioinformatics team, and a big shout out to Torsten, who's like the godfather of bioinformatics. The MIDI team, Angela and Michelle, the Root for team. And then up at MRL, Linda and Maria do amazing work with Catherine, Justin, obviously from the TB program, and then our fantastic EPIs at the Victorian Department of Health as well. So I think that that's all. I don't know if people can ask questions, but I am happy to answer any if people are able.
Well, fantastic, Kristy. I really appreciate that great talk, and I know everybody has listening in as well too. We do have time for a question or two, and I might just maybe read a composite question that has come through from the chat. So Michael Hall's question was, where does your information about level of resistance predicted come from? And perhaps just as you get that together, I'll also read Harry Walker's question out, which is of a similar area. How were AMR-associated mutations discovered or selected for the MDU custom database? So what are the criteria there for both detection and the level of resistance?
Yeah, so they're really good questions. So what we did, and this was many iterations of our validation process. So the WHO catalogue is really helpful in providing some levels of confidence for mutations. Many of the bioinformatics tools that are available, such as Microbe and TB Profiler, have extremely well curated databases that have, I'll say, reference-based mutations in them. So what we did was we actually used a composite of those databases and incorporated the WHO levels for confidence. So where there was a reference available and the WHO said, yes, this is a high confidence one, we stuck with that. There were a handful of mutations which didn't have references available. So those were sort of handled as we went with the WHO because they have a very verbose data set that they were working with. In terms of determining the level of resistance, that was actually undertaken using our validation set. So the validation set we had did have some samples that had a range of drug resistances provided. We also interrogated the literature as well. So there's about 5 mutations that are listed in the WHO database as being associated with resistance, but they also have a note that says that they are unclear. I can't remember exactly the terminology that's used, but it's in red and it's in one of the columns towards the right-hand side. And when we went back and we looked at our data, as well as the data from the literature, we could actually see that those mutations were more closely associated with low-level resistance. So resistance around that breakpoint, which is really hard to define for TB because you don't actually have a range. There's no sort of, MIC. It is resistant or it's not resistant. So determining that is challenging. Where we were unable to confirm those identifications or those calls, we erred with, again, what was in the WHO catalogue with a moderate confidence. So rather than sort of just guess, because that's what we thought, we erred on the side of going with what was in that super large data set. So it was a bit of a process. I've got some numbers, odds ratios and things like that we can talk about as well if Harry and Michael are interested in catching up to go over the data in more detail, but that's the summary.
Thanks, Kristy. That's very helpful. And I know there may be some conversations offline about many of these things. I think in the interest of time, we better wrap it up there, and I'll hand over to Ella to introduce our last speaker. But thanks again for that great presentation, Kristy, and we'll look forward to catching up at the end.
Yeah, great. Thanks, Justin. And thanks again, Kristy. That's a really excellent presentation and very impressive work that you and your team have been doing over a few years now. So, Vitali, you might want to start trying to share your screen while I'm introducing you. So our next speaker is Professor Vitali Sintchenko, who's a leading public health microbiologist and informatician who conducts research on biosurveillance and communicable diseases. He's a fellow of the RCPA and the Australian College of Health Informatics, and as the director of CIDM Public Health, he leads a public health and diagnostic microbiology team working toward improving laboratory and epidemiological investigations of communicable diseases. And he's going to be providing us with some case studies in TB genomics this morning. Your presentation looks like it's working well, so I'll let you.
Thank you very much, Ella, for this kind introduction, and thanks to organizers for having me this afternoon. I hope you can hear me well and can see my pointer on a screen. So thanks to Kristy and Chris, my job is a bit easier. The objective is to go through some case studies based on Mycobacterium tuberculosis genomics that we experienced over the years, and perhaps try to answer some of questions that both speakers have raised. I must, excuse me, I must mention that what we're talking about in terms of patient management and the implications of genomics applies to the context of a low TB incidence country with high access to diagnostic testing, high rates of culture-confirmed tuberculosis, high rates of referral of TB isolates for genomic services, availability of prospective genome sequencing and reporting to clinicians, and obviously, last but not the least, sufficient response from public health, i.e. jurisdictional TB control programs supporting contact tracing, where genomics can value add. And absence or presence of these variables are very important when you think about utility of genomics. From what Chris has already mentioned, you can infer this couple of rules. So if we have TB isolates from 3 different hosts sequenced and their genomes compared, and if genome from patient A, host A, is indistinguishable from genome from patient B, but very different with genome from patient C, it means that hosts A and B are epidemiologically related, or are likely to be epidemiologically related, and host C is not epidemiologically related to host A or host B. If we're lucky enough to have sequential isolates of Mycobacterium tuberculosis from patient A, host A, with a recurrent disease representing first and second episodes, and these genomes are sequenced and look indistinguishable, then it suggests relapse of previous disease, endogenous relapse. If these genomes are different, this finding would suggest reinfection with a different strain. So if we move to case 1, which is about deciphering community outbreaks of Mycobacterium tuberculosis. We had in New South Wales for many years large outbreak with ongoing transmission in a community. These cases have pulmonary tuberculosis. They all represented risk community, but phenotypically, these isolates are all drug susceptible to fully, and whole genome sequencing has been applied to infer potential transmission pathways. And we applied this tool for over several years, just because epidemiological links between cases were difficult to understand completely. So this is resembles what Chris was showing you a few minutes ago. Essentially, a comparison of multiple isolates sequenced by next generation sequencing methodology with good quality, and each row represents the isolate. The top row is a reference, and each column represents a position in Mycobacterium tuberculosis genome where SNPs mutations have been detected. So this is obviously not a full genome, but just selection of 39 positions in this genome. And as you can see that there are multiple isolates which are indistinguishable, really. There are no differences whatsoever. And these isolates have been recovered from patients diagnosed in different years within this outbreak. And there are also, as you can see, some fixed SNPs which define this other subclusters within this cluster at the bottom of the table. So without whole genome sequencing, when we look at the distribution of cases within this cluster, and this is a snapshot of the cluster between 2003 2013, it's pretty much everyone is connected to everyone. They know each other to some extent, to a larger extent. They've been in connection. So social network analysis gives you a very complicated picture. With whole genome sequencing on the right-hand side, you can see that this cluster is actually subdivided into two clusters with some geographical separation when you think about cases and their predominant residents. And I think Chris mentioned potentially missing cases. So this in yellow is on the right-hand side is a hypothetical node, postulated source of infection or unrecognized case, not sequenced, but there were too many SNP jumps between cases over this period of time. And you can see in a cluster, essentially there are 29 mutations within a cluster. So over a long period of time, it's more than 10 years, obviously you see mutations. But all cases are pulmonary tuberculosis, and some of these cases within cluster relapsed due to poor compliance with therapy. So I suppose we can talk about individual cases if in a bit more if there are time, if there is time for questions. Case 2 is the case about our first XDR-TB case, which was a middle-aged man with cough, night sweats, and weight loss for over 3 months, who presented to a hospital. He was of East African ancestry, spent much of his adult life as a refugee in Africa, had several treatments since his early adolescence of different duration with drugs that may be not necessarily confirmed to us. And last treatment was 3 years prior to this presentation, with 2 months of 4 drugs of therapy and 6 months of HRE, and was declared cured. So he had stable chest X-ray for several years, and because of that, was allowed to migrate to Australia with culture negative result for his sputum. On presentation, he was 3+ AFB smear positive, was immediately commenced on first-line therapy. But when we're talking about some time ago, 2014, MDR-TB plus Hain Life Science essay suggested MDR-TB. Therapy was modified with addition of amikacin and moxifloxacin at the time, pending further susceptibility results and whole genome sequencing results. We sequenced this isolate with over 5-fold average coverage, which in retrospect was excessive, but nevertheless, we really were keen to get this information accurately, and produced genomic evidence of resistance to isoniazid, rifampicin, pyrazinamide, ethambutol, fluoroquinolone, ethionamide, and injectables, amikacin, caprimycin. And on the right-hand side, you can see that there were multiple mutations associated with resistance to individual drugs. So it's not just a one drug, one mutation, or one mutation, one drug, but cumulative impact potentially is expected of mutations associated with resistance to one class of agents. For isoniazid, for example, for ethambutol, and for quinolones, for example, and injectables. So the organism was exceptionally difficult to grow. It took essentially many, many weeks to produce phenotypic susceptibility results. They were arriving in stages. Whole genome sequencing, in contrast, provided more rapid result to a treating clinician. So that was a demonstration of relevance for guiding treatment at the time when obviously 3-4 active drugs were required to prevent amplification of drug resistance to further levels. So that was drug resistance story that at the time changed perceptions of treating clinicians about whole genome sequencing information. It was a very important milestone. Case 3 is about laboratory contamination events, which whole genome sequencing can identify and assist with investigation and action. And Justin was involved in this case as well, and I hope Justin remembers this case and could comment further if needed. So we had 2 isolates, one isolate from patient 1, who recently migrated from high incidence country to Australia. TB was recovered from sputum and BAL, was positive on expert ultra for TB and rifampicin resistance, and whole genome sequencing produced MDR-TB resistor, and Beijing lineage was identified. Treatment was modified according to lab results to bedaquiline, amikacin, pyrazinamide, and clofazimine. And we also learned subsequently that this patient has had close relative treated for MDR-TB in Nepal. So on the right-hand side, you have a phenotype and resistance for this isolate. Resistance to isoniazid, rifampicin, ethambutol, streptomycin, moxifloxacin, and kanamycin, ethionamide. So it's one mutation per class of drug, and I think it was quite useful information at the time for managing therapeutics for this patient. But we were receiving another isolate for patient 2. So patient 2 lives in another state, received a standard therapy for lymph node TB in another high incidence country, some time ago, and his discharging swab, his discharging lymph node swab grew Mycobacterium tuberculosis, which was positive in the diagnostic lab on expert ultra PCR as well. So genome sequences of isolates from patient 1 and 2 were genomically indistinguishable. So this is where investigation started. And so what we've learned that both isolates were referred to our laboratory in Sydney from the same diagnostic laboratory, and was actually interstate diagnostic laboratory. Not that it matters. It was just first in the middle of pandemic and exhausted, as you would imagine, and this timeline, and we published eventually this, and Alice Gordon, our postdoctoral research fellow in genomics, led this short publication, puts this graph together, explaining the timeline between collection of samples, the diagnosis, and WGS report. What is important that specimens were collected around the same time from these two individuals. They're processed on the same day in a diagnostic laboratory. Specimen for patient 2 flagged positive by immediate within kind of 3 weeks. It took a lot longer for specimen from patient 2 to become positive. And specimen was smear negative, while patient 1 had sputum and then BAL positive for culture, both of which detected MDR-TB, and subsequent TB biopsy sample for patient 2 remained culture negative. So it led to identification of this probable lab contamination event, modification of therapy for patient 2 from MDR therapy to standard therapy, and I really hope that it worked for patient 2 and patient 1 together, and they've been cured successfully. Case 4, and Justin, please stop me if when needed. If we're running out of time. So this is phenotypically susceptible, genomically resistant Mycobacterium tuberculosis isolate recovered from a case with advanced cavitary disease, history of previous therapy, phenotypically, and this was before the criteria, the actual breakpoint concentration for resistance to rifampicin was revised. We reported fully sensitive organism phenotypically. Experts gave RPO positive resistance result, and assay produced negative, fully susceptible resistance profile. So it was discrepant result between two molecular assays. Whole genome sequencing recovered mutation in KET-G gene, which was unusual mutation, and not included in Hain assay, and RPOB gene mutation just to the margin of rifampicin resistance determining region. And MIC was really borderline. Interpretation was for genomically MDR-TB, and our clinicians treated this as MDR-TB rather than phenotypically susceptible organism as far as therapeutic choices are concerned. In mind, because we obviously been aware of this occult MDR-TB cases with higher risk of recurrent or persistent disease if normal therapy is concerned. I mentioned the capacity of whole genome sequencing to differentiate between exogenous reinfection and endogenous relapse, and this is a study that we published some time ago with our colleagues in India on 2 sets of patients with a recurrent disease. One set of HIV negative patients and another set is HIV positive patient set. And you can see the difference between these cohorts. In HIV negative cohorts is all kind of little pairs of genomically indistinguishable isolates recovered from individual cases. Some of them have been associated with resistance to isoniazid. Some of them developed rifampicin resistance in a recurrent episode. But essentially, it's all endogenous, predominantly endogenous reinfection. In contrast, in HIV positive cohort, there are patients that have been reinfected with other cases of Mycobacterium tuberculosis in a community circulating at the time, and it is clearly seen that you can there are a lot more unique sequences, not paired sequences, from same cases diagnosed with recurrent infection. So the rate of relapsed infection obviously is one of the public health TB control measures that we're interested in. It remains very low in Australia and in New South Wales. When you apply whole genome sequencing capacity for this sequenced relapses, you can see that only 13% in our latest study were really reinfections. Majority were endogenous relapse due to problems with compliance, etc. So it's not really, it's a clinical management issue rather than public health issue. I'm mindful of the time, Justin and Ella. Maybe I should stop. I've got 2 more cases, but probably I should stop.
Oh, I think that's been fantastically illustrative, Vitali, of so much work that you and the rest of your team are doing. And knowing that there's more cases to come, I think probably teases us for next time as well, but we'll look forward to it. Ella, would you like to say a few things as we come to the end here?
Yeah, thanks. Look, thanks so much to all 3 of our speakers for providing sort of really different and excellent sort of overviews of their various topics. There's so much more we could talk about today. I'd also just like to thank particularly Tanya Mukherjee and Elise Guevara-Rattray for their excellent assistance in organising today's webinar, and Justin for agreeing to co-chair with me. So thanks so much, everybody, and I think we'll wrap it up there.
Fantastic. Thanks very much, everybody.